Okay, this is the Professor, CBMD PhD, on today's Tuesday, May 16th, 2023. It is 5:36 and 17 seconds, a.m. Eastern Daylight Time, and today's research, human DNA. So let's hop to it here. Human DNA is everywhere. That is a boon for science, and an ethical quagmire. Date May 15th, 2023. Source: University of Florida. Human environmental DNA is ubiquitous in air, soil, and water. Samples can be traced to individuals, demonstrating new scientific possibilities, but raising ethical concerns around consent with eDNA research. We cough, spit, shed, and flush our DNA into all these places, and countless more. Signs of human life can be found nearly everywhere, short of isolated islands and remote mountaintops. That ubiquity is both a scientific boon and ethical dilemma, say researchers who sequenced a widespread DNA. The DNA was of such high quality that the scientists could identify mutations associated with disease and dilemma with genetic ancestry of nearby populations that could even match genetic information to individual participants who had volunteered to have their errant DNA recovered. The researchers say that ethically handled environmental DNA samples could benefit fields from medicine and environmental science to archaeology and criminal forensics. And for example, researchers can track cancer mutations from wastewater or spot undiscovered archaeological sites by checking for hidden human DNA, or detectives could identify suspects from the DNA floating in the air of a crime scene. As this level of personal information must be handled extremely carefully, now scientists and regulators must grapple with the ethical dilemmas inherent in accidentally or intentionally sweeping up human genetic information, not from blood samples, but from scoop of sand, a vial of water, or a person's breath. They've been consistently surprised throughout this project at how much human DNA they find in the quality of that DNA. In most cases, the quality is almost equivalent to if you took a sample from a person. And because of the ability to potentially identify individuals, the researchers say that ethical guardrails are necessary for this kind of research. It is a standard in science to make these sequences publicly available, but that also means if you don't screen out human information, anyone can come along and harvest this information. Researchers successfully used environmental DNA, or eDNA, to study endangered sea turtles and the viral cancers they are susceptible to. They plucked useful DNA out of turtle tracks in the sand, greatly accelerating the research program. The scientists knew that human eDNA would end up in their turtle samples and probably many other places they looked. With modern genetic sequencing technology, it is now straightforward to sequence the DNA of every organism in an environmental sample. The questions were how much human DNA there would be and whether it was intact enough to harbor useful information. The team found quality human DNA in the ocean and rivers surrounding the Whitney Laboratory, both near town and far from human settlement, as well as in sand from isolated beaches in a test facilitated by National Park Service. The researchers traveled to a part of a remote island never visited by people. It was free of human DNA, as expected, but they were able to retrieve DNA from voluntary participants, footprints in the sand that could sequence parts of their genomes with permission from the anonymous participants. They also tested a technique in native Ireland, tracing along a river that winds through town on its way to the ocean. They found human DNA everywhere, but the remote mountain stream where the river starts, far from civilization. The scientists also collected room air samples from a veterinary hospital that recovered DNA, matching the staff, the animal patient, and common animal viruses. Now that it is clear human eDNA can be readily sampled, researchers say it is time for policymakers and scientific communities to take issues around consent and privacy seriously and balance them against the possible benefits of studying the errant DNA. Any time we make a technological advance, there are beneficial things that the technology can be used for, and concerning things that the technology can be used for. It is no different here. These are issues they are trying to raise early so policymakers and society have time to develop regulations. Good day. This is the Professor of Microbiology, Bioinformatics, Pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.